to wade into this topic, I want to put this into the context of how cellular machines in general might evolve, and the synapse, of course, is one such cellular machine. Their cells have lots of them: proteasome, nuclear pore, spliceosome. There are lots of them. And we know a lot about the component proteins of these machines. This is the proteasome right here with 32 proteins. But the evolutionary driver for a cellular machine is most likely to be protein-protein interaction domains. These things have to be assembled, and if there are 32 proteins in a machine, it's likely that at some point there is selection for every single one of those interactions. And they may be happening collectively in some way, but these machines are not, obviously, over evolutionary time, not assembling all at once. So if we want to try to break that down a little bit and look at this evolutionary process, that's been the subject of interest for my lab. And it's the synapse that has been what's interesting to us. So if one is interested in the origins, the evolutionary origins of the synapse, and here is a way of looking at just a generic chemical synapse with a presynaptic side here with synaptic vesicles and scaffolding proteins that hold these things in place. The adherence junction on either side of the synapse, which is keeping the pre- and postsynaptic compartments in register with each other, so molecules don't diffuse away in the synaptic cleft. And then a scaffold layer on the postsynaptic side that's holding the receptors and channels in place. And all of these are very close to the surface of the synapse. It's not till you go deeper into the synapse that you have these more generic components like the cytoskeleton and cellular signaling molecules that are then communicating down to the nucleus. So we have here a generic synapse. And these structures appear early in animal evolution, but not at the very origins of animal evolution. They really, you first begin to see cell organisms with synapses and neurons here in Cnidaria. Also, ctenophores have nerve nets, and of course, Bilateria. But sponges, here also a very basal organism, it completely lacks neurons, synapses, or anything remotely connected to a nervous system. So it's this evolutionary transition right here where it is likely that synapses emerged, if, as we heard last night, if you're of the cladistic beliefs, which is the approach that I'm taking. It's always formally possible that nervous systems were lost at an earlier time point. So sponges lie right here at near the root of the animal tree, and it's believed that the single cell organism that's in evolutionary terms work from the coal king is closest to the animals are the choanoflagellates. There's thinking that's emerging now that may be changing that a little bit, but we'll be talking about relationships of organisms that go from here all the way up to here in this presentation. So here are some features of sponges. They have no true tissues or organ systems. They have a set of cells that we know about, but those cells have not come together to function as an organ system. So it's not just it's not that they don't have nervous systems only. They don't have a gut or many other organ systems that other animals have. A limited number of cell types. It's a very successful species, over 10,000. A very successful phylum, over 10,000 species. Wide range in size, and again, depending on who you believe, if you really sort of push the fossil record a little bit, you can say that the fossil record goes back about 600 million years. This here is the embryo of a sponge, and then these are the adult forms. Here are the major cell types that you see listed here. You can see one of them is called a choanocyte, which is thought to have been from the choanoflagellate lineage, as this cell has almost been wholesale incorporated into the anatomy of a sponge. There's a lot of morphological similarity. And but even though sponges don't have a nervous system, they are able to function with some degree of coordination of their cells. So here's this spongiocele in the center and these ostia where water will go right through like that. And for water to pass through that water column in that synchronized way, you see these little cilia here. There has to be some coordination among these cells, which is still poorly understood. Okay, so now what we did with that introduction, what we did was to sample when these studies started, and I'll talk about more data as the talk goes on, but we started just with a rather sparse but broad sampling of the animal kingdom here and looking at these different species. Both the sponge, for which we now have a genome, the Amphimeda queenslandica, a cnidarian, Nematostella, which we have a genome, a Drosophila, and humans, and to pull out from all these species with nervous systems a set of synaptic genes, and then to search for those same genes, whether or not those genes exist in an organism that doesn't have a nervous system. And to our surprise, there were lots of synaptic genes in the sponge. So, and here is, in this cartoon, you can see here in blue are the various, this is just the postsynaptic region. Here's the scaffolding proteins. Here are deeper connections to the cytoskeleton. Here are the channels and receptors. And those that are in blue are all encoded and expressed in the sponge. These are all synaptic genes that are encoded and expressed in the sponge genome, and these few up here are not. A few that are not are very, very important. For instance, the NMDA receptor does not exist in the sponge, but there's a large number that are present in the sponge, and most of them seem to fall right along this scaffolding layer. They're very, very ancient, and up here, some of them along the plasma membrane. So the sponge has something that is has components of what we might call a proto-synapse. Now, I say that they're expressed, and we actually know that all these genes are actually expressed because in a few slides further, I'll talk about doing transcriptomes on the sponge. But at the time that we first published this, we also took a representative sample of some of the genes that are expressed in sponge, and they're also in synapses, and did in situ hybridization. This part is in collaboration with Bernie Degnan at University of Queensland, and showed that by in situ hybridization, all of these genes were expressed, number one, but importantly, they were also all expressed in the same cell type, called a globular cell, here along the surface of the larval form. So the next question would be, is that if all these genes are expressed, then are they also assembling with each other into something? And we don't know the answer for all of them. We don't actually know the answer for any of them, but we have some good evidence that at least one interaction that occurs in organisms with a synapse is also occurring in the sponge. And the way we did that was to look at a relationship, a PDZ binding relationship between one of the components, one of the scaffolding proteins, which is called PSD95, and it's made up of several domains called PDZ domains, and inserting into that PDZ domain, in this crevice here, is another protein called crypt. So the crystals, the co-crystal has been, we have all the coordinates for the co-crystal. And what we did, this was done in collaboration with Bruce Tidor at MIT, we were able to thread this sponge sequence into the known crystal structure and actually assign which amino acids were conserved and which were not. So CRIP here is the ligand, and this whole globular structure is the PDZ domain, and all these gray amino acids have about a 50% conservation between sponge and human. However, those amino acids here in blue, which are actually binding to the ligand, are 100% conserved between sponge and human. From that, we inferred, but as I say, we haven't proven yet, that this interaction, at least this interaction, is very likely to exist in the sponge. So we concluded, using this concept that has been discussed by Stephen Jay Gould, this concept of exaptation, that and here's the definition, which you already know: biological unit originating with and selected for a function other than that for which it was later selected. And we believe that we have an example here of what might be called molecular exaptation, in which a set of genes, possibly assembled together, were in the sponge, were exapted to become to be used to begin to lay the groundwork for creating a synapse. Now, we've expanded this work quite a bit, and I showed you the presynaptic side, the postsynaptic side. Here are all the proteins that you see from the presynaptic side here of a human synapse, with the synaptic vesicle here as it's maturing, scaffolding proteins along here, and this is pretty much what we know about the presynaptic side. So let's do the same sort of pattern and see what's there, how these different protein components of the presynaptic side were added over evolutionary time. And if you start with yeast, there's very little. There are certainly yeast have to engage in vesicle fusion, and some of the vesicle, synaptic vesicle associated proteins are present, as you can see here, with where fusion occurs, but there's not very much that's here. If we go to monocyga, which is the choanoflagellate that was completely sequenced by Nicole King, you can see here that there's still very little. There's a bit more, but I don't think we could ever say that anything here looks remotely yet like a presynaptic element. Now here's the sponge, and now we're getting pretty close. Again, it's very much like the postsynaptic side, where many of the elements here to make a presynaptic part of the synapse are now present. There are some, again, key things missing. These genes, piccolo and bassoon, don't come in until very late in evolution. But on this side here, we really have an awful lot of the function is already in place. And when we get to cnidarians now, which do have the nerve net and a presynaptic side, things are pretty much in place. As I mentioned, piccolo and bassoon don't show up until we move into 3 germ layers. We move out of cnidaria and ctenophores. Okay, until we move into bilaterians. Okay, so let's go a little deeper now into this PDZ domain, which I discussed, because the PDZ domain is a key structure for scaffolding receptors within the synapse. And so that really would be a nice place to start, because a lot of things hang off of that, and it could be a good place to sort of originate a synaptic synapse, the components of a synapse. So the PDZ domain is about 80 to 90 amino acids, and it's a very versatile motif. It's been around for a very long time. I'll show you that in a moment. That connects many proteins in the postsynaptic junction. As you already know, we have a crystal structure for the PDZ domain, and the new information on this slide is hidden down here in the corner, where when a ligand binds to a PDZ domain, it binds with about 4, 5, 6 amino acids at the extreme C-terminus of the ligand. And the characteristic way in which you recognize a PDZ binding protein is with a hydrophobic, usually a valine, as the extreme C-terminal amino acid. And then there's other parts of the consensus sequence. For instance, this position right here is almost always a serine or a threonine. These class 1s are the most common. So we have a characteristic, we have a motif for the ligand, and we know a lot about this PDZ domain. So let's go a little deeper and look at that. And to go a little deeper into evolutionary history, let's take a look now at this creature, which I've already introduced. This is a choanoflagellate. Some people would say perhaps one of the closest single-cell organisms to the animal kingdom. And when you look into the choanoflagellate genome, what's really remarkable about the choanoflagellate genome is the explosion of domains within proteins that occurred at this time in evolution. That is, domain expansion really preceded the origination of multicellularity, unless there's some attempts here at multicellularity, but really what, and I think multicellularity is really not the right word to use. Really what we really want to say what happened at this juncture as multicellular organisms such as animals and plants evolved is not so much the multicellularity itself, which evolved many times, but cell specialization, which is really the key. So, so here is this right here, the monocyga. Here is the M, that's the choanoflagellate, and there are other places where domains clearly expanded. Here's a whole bunch of domains, and their numbers at different times in evolution. You can see here Neurospora and Dictyostelium. Very, very few of the domains we recognize that are the building blocks of multicellular organisms. But here you can see that in these organisms here, Nematostella and as early as Monocyga, there is this large number of domains, including the PDZ domain. So PDZ domains expanded greatly here at this time, around the origination along some ancestor of Monocyga and all Monocyga and animals. So now if you go a little further back, and you again look at this postsynaptic element, what's present back in the choanoflagellates? Well, now almost everything is gone, but there is a very ancient gene present in Monocyga here. This PSD95, it has lots of names, DLG, which is made up of these PDZ domains. This is the exact same scaffolding protein that is present in human, holding all the receptors in the postsynaptic element, as I've already mentioned, and it's got a very close homology between choanoflagellates and even humans in terms of the organization of this of the domain structure here. Now, one thing that's quite interesting is that you can see a lot of so what's in blue are the choanoflagellates, and so all these other colors are not present in choanoflagellates, but there are some of the ligands that will become ligands for PSD95 are also present in choanoflagellates, but the actual that consensus motif is not present in these ligands. That is, these proteins, these genes existed in choanoflagellate, but they could not bind to this scaffold because the motif at their C terminus is not present on these proteins. So we have the scaffold, we have a few of the elements that will become involved in a synapse, but this, to me, does not look like it's assembled into anything. Now, I want to now introduce a concept here, which is about the clade of PDZ domains. As I said, here's that PSD95 or DLG scaffolding protein. As I already mentioned, it has these three PDZ domains. It has other domains too, SH3 domain, GK domain. There's other domains in this protein that are highly conserved even in Monocyga. But the point now about clading is that PDZ domains are present in many, many different proteins. So here we have, as we look at the first PDZ domain of this protein called PSD95, as we look at it, this represents a clade. It's PDZ1 of the PSD95 in species that range from humans to choanoflagellates, and PDZ2 and PDZ3. And then you can go on to other genes like Cas, and here's another PDZ domain that might potentially clade quite differently than the 3 PDZ domains in PSD95. So when we talk about expansion of these PDZ domains, what that means is that they've wound up in all these different settings. Here's one here, there were 3 in PSD95, and they have these cladistic properties here of having a lot of homology in this direction and less in this direction. So human PDZ1 is much more like sponge PDZ1 than it is like human PDZ2. Okay, now PDZ domain evolution, here we take another term from that I think Stephen Jay Gould was the first one to say, this long fuse explosion property, in which organisms existed for a very, very long time, all these single cell organisms, and then here's monozyga, and the lines in this direction are the number of PDZ domains, so we have very few here, and then we have this explosion here in monozyga of PDZ domains. And this, you can get these patterns for a lot of different domains. So they would not look identical, but this is for the PDZ, and then they're increasing, and there's another explosion of PDZ domains here when we get into vertebrates. So so we have this sort of long fuse where there's millions of years of evolution going on. There's this burst in PDZ domains, and then another burst in PDZ domains. So give me a moment to introduce this next slide. The question is, if monozyga has this scaffold for a synapse, but yet synaptic proteins are not binding to it, what is binding to it? So one of the experiments we did was to then do what's called 2-hybrid, which is to clone the tri-PDZ domain, the 3 PDZ domains from monozyga, PSD95, all 3 of them, and use them in a 2-hybrid system to fish out the interactors with that domain. And this is what we got. We got all these very strange beasts. We hardly recognize any of them. They're just they're proteins that certainly have nothing to do with a synapse. They really were. It seems like in monozyga, whatever this scaffolding structure is scaffolding has a very, very different function. But they look at that. All of these strange proteins have this hydrophobic, usually a valine at the C terminus. So the PDZ domain is acting like a PDZ domain. It's just that it's binding to a lot of other interesting stuff. So now the next thing we did was to collect all the PDZ domains. At first we just did bilateria. All the PDZ domains of bilateria, and line up 76 amino acid positions, and look at their conservation all through all of them. There are certain positions across the 76 amino acids which are absolutely conserved 100% from monozyga, or certainly from sponge, all the way to human. Those we call globally conserved. There are other positions which are only conserved within a clade, so that position is only similar, say, in PDZ1 of PSD95. That position is the same in sponge through human, but if you go to PDZ2, now it's changed. So this is, here was our definitions here. So we have globally conserved positions. Here's an example of 2 of them. We have neutral positions that they didn't care where they were. And then we have these classifying positions, because these were positions that tells you what the clade is. If you have a methionine in some position, you can say, that PDZ domain must belong to PDZ2 of PSD95. So if you now graph this and you look at put global entropy or global conservation on one axis and the within clade conservation on another axis, and you look at how the correlation, it turns out that there are 6 outliers. They just don't fit here along this line. These outliers mean that they are showing that they have they're within clade entropy is falling off the line compared to their global entropy, which means that they could be classifying positions. These are the positions. These 6 positions tell you which clade you're in. So when PDZ domains expanded cladistically, these were the positions that evolution chose to conserve. So what's important about these positions? And so here they are. These are the 6 classifying positions in bilateria, and they can predict which clade you're in with 96% accuracy, just those 6 positions. And let's see a little bit more about them. So this is, here they are. So the next question would be, well, if they predict the PDZ clades so well in bilateria, how well do they do in sponge and cnidarians? They do pretty well. In sponge it goes down to 77%, but it's not bad. In cnidarians it's 83%. All of invertebrates 86%. But here you lose it. So although Monocyga expanded in its PDZ domains, it did not expand in the same cladistic pattern that animals did. There's no way to predict the domain here. Now, of those 6 positions, where would they lie? Where do they lie in the PDZ domain? And so 4 of the 6, not surprisingly, lie in all these different proteins that have PDZ domains. Syntenin, shank, these are all proteins with PDZ domains. Those 4 of the 6 positions lie here very close to the ligand. They actually touch the ligand. 2 of the positions are on the complete opposite side of the molecule. And we don't understand why these positions are conserved among clades. It's not at all clear yet. So we now think that in terms of the evolution of the scaffold, we have this tri-PDZ domain present in Monocyga. And it looks like a PDZ domain. It binds the motif. But it's not till we move into sponge that we start to get fixation of classifying positions, and then we get this tremendous expansion. So last part of the talk is I now want to go and tell you the most recent data that we've done. We've now completed transcriptomes on every life cycle 4 life cycle stages of the sponge here. This is material that was collected from Great Barrier Reef off of Heron Island by Bernie Degnan. And in looking at all these from these pre-competent larvae here to that settle and become competent, and post-larva about to metamorphose into adults, we did transcriptomes on all these stages. And we looked at the expression of the synaptic genes in the transcriptome now at each of these stages. So here is, again, another way of looking at the synaptic network. And here's the presynaptic elements, postsynaptic, it's called DLG4 here, or PSD95. Here's the synaptic cleft, and here's the postsynaptic side. So, and those that are in red are present in the sponge. So again, same point, a lot of them are present. But now what we can do is we can now look at the, over time, we can now watch these patterns of expression for each one of these synaptic genes. And in so doing, and we can compare it to all of these other animals that have synapses, and corals and cnidarian, and then all of these other bilateria. So what becomes apparent here in these matrices and looking at a network analysis is that first, there is this modular quality to expression. There are certain sets of genes that seem to cluster. And in a way that is sort of apparent here, but is very highly statistically significant, these modules in the sponge are really quite isolated. That is, there's cool colors, blue, that surround them, and they don't really seem like especially these, that they're very highly interconnected, in contrast to all these other conditions where surrounding the modules are warmer colors, suggesting we've shown much higher connectivity. So at the transition from sponge to all these other animals, what happens is that the modules still exist, but what happens is that we now have modules that may be functioning quasi-independently in the sponge are beginning to get wired together, and this very discrete modularity here tends to diminish. So modularity is decaying and connectivity is increasing. And if you now want to look here at these are these are all these genes and synapses and these different creatures, and the color coding here is has to do with which one of these modules they belong to. And when we do this, it turns out that more than 50% of genes belong to the same co-regulatory module. The way the clustering is done, the network analysis is done to create the modules, has no bias. We set up these patterns of expression, and these modules emerge. You could then ask, well, what is driving these communities, these modules? Well, the modules seem to have at least some functional relevance because more than 50% of the genes belong to the same these genes on here, where these circles are, belong to the same co-regulatory module. And the one that's the strongest is this VATPase. That one is always very modular. It's this thing right down here, which is these genes always collect in a module by themselves. So lying seamlessly within a synapse is an ancient modular architecture here that you can still see and seems to be, again, exapted from the sponge to now form a coordinated functioning synapse. And finally, just to do some comparisons, we didn't look at, it wasn't like the synapse was the only machine we looked at. We looked at a proteasome, which I'll show you here, and a nuclear pore, and we looked at some others that were more complex structures, like epithelial type structures, and did a lot of comparisons. Here is this is all the proteins in a proteasome, and compared to the synapse, this is much, much more interconnected. That is, every one of these proteins, which would be considered a node, has many more connections. So that's called, in network speak, that would be the degree. And you can see here that synapses here are of a very relatively low degree because they don't have nearly as many connections to others as the as the nuclear pore here or the proteasome out here, which is very, very highly connected. So this is a little hard to look at. So, but we can just sort of zero in down on some of these control machines that although here in coral and in sponge, the proteasome, the nuclear pore, are just broken up more into modules, it still has a lot of interconnectivity. So we believe that cellular machines that are very, very ancient, those that existed in single-cell organisms, are already highly interconnected in sponge, in contrast to a synapse in which interconnectivity of ancient modules is going to happen later. So here are the conclusions for the talk. The evolution of the nervous system required that novel binding partners compete with pre-existing ligands in a manner that does not disrupt the assembly of a protocomplex. What that means is that here's the sponge scaffolding domain. It's got a lot of stuff that we recognize in a synapse binding to the PDZ domain, but it doesn't have an NMDA receptor. How's the NMDA receptor, when that gene is invented, going to now come along and compete with the existing ligands on the PSD95? It has a little bit of a job to do. There's different ways in which that can be done, and one way in which it can be done, perhaps, which I'll get to in a moment, is by simply increasing the number of PSD95s that you make. Make more of them so that you don't have to compete as hard to bind. So regulation, which is implied in all those pictures of the networks, is a key feature, I think, in the evolution here of the synapse. In the case of PDZ domains, fixation of domain order preceded the establishment of classifying positions. Monosega already has the 3 PDZ domains. So there's syntony between the PDZ domain order of Monocyga and the rest of the animal kingdom. But what Monocyga lacks is any kind of key positions which are now resulting in these clades of different types of PDZ domains. Relatively minimal modifications of the core modular structure of, say, a sponge protosynapse, can result in functional evolutionary leaps. It only required a few more genes to go from what the sponge has to nervous systems and synapses as we know them. Evolution by regulatory elements, I think this is implied in a lot of the network stuff I showed you. Upregulation of scaffold protein expression represents a possible solution to this issue. I already said that. Synaptic gene networks have a modular pass detectable by co-regulatory gene expression networks. So there we have it. And see how the time's doing? I think I have 1 minute to show you a little bit of a lighter slide. Because after I gave this talk, at MIT not too long ago, somebody who I don't know posted this on his website. This says, he said that these were the dreams of sea sponges. The talk was about the dreams of sea sponges. So I discussed this with one of my colleagues. I said, somebody posted this blog about the talk, and they had this very attractive title for the talk. And he said to me, well, what is the sponge dreaming about? So sponge is dreaming about getting a brain. Here's our group. Early on, we had collaborations with Bruce Tidor and his student Catherine Armstrong at MIT. We worked with Mansi Srivastava, who really was the first author on the whole genome sequence of the sponge. Bernie Degnan and Sandy Degnan and people who did the in situ in his lab. Amphimedon, a key contributor to this project. And here in my group, the first person who took up this project was owner Sakara, right there. And he then handed the project over to the network analysis to Cecilia Kanaka right there, and Danny Bissett, who's not shown. Todd Oakley, who maybe some of you probably know, really helped me a lot to learn a lot about evolution and was very helpful early on with this project. Mary Arcila, who's here actually right there, did all the sequencing, and Robin Zhu did the computation as the data came off the sequencer. Thank you.